Hepatocytes, the pale cells in this cartoon of a liver lobe, are the liver's parenchymal epithelial cells. They support the two principal functions of the liver. The first is filtering blood coming from the intestine, which is processed by hepatocytes before joining the general circulation. To mediate this filtration, hepatocytes organize in single-cell cords that face blood vessels on either of two basal domains. The second function is sending bile, a detergent-like substance, to the bile duct for transport to the duodenum via a bile canaliculi network, here shown in pink, which is formed by hepatocytes' apical domains. The molecular exchange with the blood is facilitated by the lack of a basement membrane and the presence of only sparse extracellular matrix in the space between hepatocytes and blood vessels. In common liver diseases, however, this space becomes occluded by deposition of collagen and laminin, impeding the exchange between hepatocytes and the blood. Because fibrotic liver disease is also accompanied by distortions of the bile canaliculi network, we investigated if an increase in the rigidity of the extracellular matrix is associated with collagen deposition impairs the integrity of the bile canaliculi network. To that end, we adopted a protocol for the culture of primary rodent hepatocytes developed in the 80s by Martin Jarmusch's group and others, in which isolated hepatocytes are plated on collagen gel and upon attachment overlaid by additional collagen, mimicking the in vivo situation in that the matrix is present at both basal domains of the cells and the matrix rigidity is low. The polarized cultures form elaborate bile canaliculi networks outlined by the tight junction marker 01 and marked by active myosin and F-actin that are entirely restricted around the contractile bile canaliculi. We varied the rigidity of the plating matrix by comparing cultures on collagen gel with those on collagen-coated glass or on collagen-functionalized polyacrylamide gels with rigidities of 0.5 or 20 kilopascal. The substrates of higher rigidity, glass or 20 kilopascal hydrogel, featured stress fibers, as well as a less continuous bile canaliculi network than those apparent in the cultured on softer matrix. We quantified the bile canaliculi integrity as bile canaliculi length fraction, shown here in the graph on the right, the ratio between the length of bile canaliculi and the length of cell-cell contact. Because we provided collagen for a matrix, we hypothesized that inhibiting relevant collagen receptors would rescue the bile canaliculi network. We focused on hepatocytes' only integrin-based collagen receptor, alpha-1 beta-1 integrin. However, when we inhibited alpha-1 beta-1, we found the opposite from what we expected. Instead of improving bile canaliculi formation on rigid matrix, depletion of alpha-1 or a function-blocking alpha-1 beta-1 antibody disrupted the bile canaliculi network further. Notably, alpha-1 beta-1 inhibition caused the appearance of substrate parallel F-actin bundles across the overlay surface, and these F-actin bundles were flanked not by integrins but by cell-cell adhesions, here shown as pointed by the arrowhead for beta-catenin. Based on these and additional data, we suggest the following working model. On soft matrix, contractile F-actin is restricted around bile canaliculi. Cells respond to increased matrix rigidity with traction forces that then cause increased cell-cell adhesion tension. Alpha-1 beta-1 contributes to maintaining cell-cell adhesion forces, which in hepatocytes are oriented perpendicular to their basal domains. In the absence of alpha-1 beta-1 and with exceedingly higher rigidity, cell-cell cohesion can only be maintained when the adhesion junctions are organized substrate parallel, an organization reminiscent of that in classic duct-forming epithelial cells, and which leads to the loss of bile canaliculi. It's incompatible with bile canaliculi maintenance. Because alpha-1 beta-1 itself is colocalized with adherent junction along the lateral domains, in future work we will investigate if it regulates adherent junctions directly. If you want to read more details about this study, please go to our open access paper in Journal of Cell Science, and thanks for watching.